I am just going to change tack a little bit and talk a bit about a resource that the Cloudworthy Lab has been building for the past several months, and we've now made it completely publicly available on CNS-border-cell-atlas.org. And I'm just going to talk a bit about it, then give you a short demonstration. And if you've got any questions, please feel free to reach out to us. So as mentioned by David, we're on Twitter, this is our website, so do reach out to us if you're interested in the work we're doing. And just before I go any further, I'd just like to give a special thanks to the cell gen team at Sanger, especially Martin, who's really helped me with creating this resource. So what is CNS-border-cell-atlas.org? So this entire endeavor started with this review paper that David was just presenting. And it's meant to be an interactive, public and accessible tool to explore gene expression and pathway enrichment, and also perform DG analysis at a single cell resolution. So what we did was we, at the time, which was somewhere in the middle of 2022, we looked for data sets with published single cell RNA sequencing data off the CNS borders, or the meninges, which has been the theme of today. And we integrated all of these tools using SCPI, which is basically the state-of-the-art a model-based tool for integrating data sets. And what we have done in this version of the atlas is basically subset the cells for cells under homeostatic condition. So everything here represents control cells, and as you can see from this UMAP in the center over here, we see a nice representation of many different cell types, including B cells, which David has been talking about, but also T cells, macrophages, like Vivian was talking about, etc., etc. So a very good representation of different cell types, and also quite a decent cell number with 130,000 cells in total. And the rationale of creating this atlas initially was to really create a comprehensive and high-dimensional overview of immune cells at the CNS borders. And we also know that combining multiple data sets gives us more cells, which in turn reduces biases that may be associated with the independent studies involved or included here. Also, we do know that more cells gives us more biology, such as, for example, the ability to uncover more uncommon or rarer cell types. So the CNS Border Cell Atlas portal is meant to be a public resource in the spirit of open science. There's absolutely no coding required, so it's highly accessible. And also in the spirit of sustainability, it's meant to reduce, reuse, and recycle these very costly experiments that have been put into curating all of these data sets. So in the interest of time, I'm not going to go through the methods today, but in terms of future plans, we really hope to be improving the cell type annotations, because currently it's quite broad, include and start building on disease state atlases, because it's quite interesting to think about how these compartments are perturbed in disease versus control, and also start building some human atlases of human meninges. So before I go into a demonstration, once again, if you are interested in working with us on this, if you're interested in knowing more about what we do reach out to Mena, whose email is down here in the bottom right corner, or myself. Right, so I'm just going to stop my screen sharing here and click on this link, and that will take us to hopefully this web page right here. Okay, can everyone see my screen? Brilliant, thank you. Right, so when you click on CNS-border-cell-atlas.org, it comes to this landing page with a short summary of immune cells at the CNS borders, and the paper that David was just presenting here on the right. But what you really like to do is click on this, which is a viewer, and that takes you to this web page over here, the open interactive viewer. Now, as you can see here, once you come to this website, you are presented with this UMAP plot over here. Now, on the left over here, you have all the metadata that's available, whether it's continuous fields of metadata or categorical metadata. So, for example, if we expand on cell type, I can color it by cell type with the legend on the left over here. And if I click on this icon over here, I can just simply overlay these cell type clusters on top of this. I can also color by site, for example. So this is information on where exactly these cells were found from the original studies. So just going back to the cell type, in keeping with the theme of David's presentation, let's focus a bit more on B cells and plasma cells for today. Right, so just to prove to you that this top cluster here are B cells and are indeed B cells, let's plot CD79B. And if you can see this over here, they light up really nicely for CD79B, proving to us indeed that these are B cells. And if you highlight J chain, you can see that they light up for this cluster over here, showing to us that these are plasma cells. So we've decided that these are the annotation space in these genes that are expressed over here. Now, just to, for those of you that are sharp-eyed, you would have noticed that the legend here is actually inversed at the moment. So we're working on fixing that, and hopefully that will be pushed out very, very quickly. But let's focus on the B cells, as I've mentioned earlier. So what we can do is if you click on this select tool over here and CD79B selection, I'm going to just trace out the B cells over here really nicely. And I'm going to subset for these cells by clicking on this button over here. I'm going to drag these to the center and zoom in on these B cells. Now, one of the things that David was talking about earlier was that there is some B cell heterogeneity in the meningeal compartment. So just to illustrate that with some of the genes he's mentioned earlier, let's type in a few genes of interest. So IL-7 receptor and RAG1. So both IL-7 receptor and RAG1 are known to be more expressed on immature populations of B cells. So if I color that in, you can see that RAG1 is expressed over here on the left, and so is IL-7 receptor. Conversely, more mature populations of B cells tend to upregulate CD20, which is encoded by MS4A1. I color that in over here. It lights up more on the right side of this UMAP, and also H2AA, which is a class 2 MHC gene. So immediately you can see that there's some heterogeneity in this B cell compartment, with the more immature B cells perhaps sitting more to the left and the more mature B cells sitting to the right over here. Now, one of the questions we think about a lot as a lab is what exactly are the factors that position the B cells in the meninges. And one of the obvious things to think about when you think about factors positioning cells in the meninges are the chemokine receptors. Now, as David's mentioned, some of the chemokine receptors associated with B cells include CXCR4 and CXCR5. So I'm just going to plot that out real quickly: CXCR4 and CXCR5. Now, what you notice over here is that if I click on CXCR4, it seems to be expressed both on the more immature populations of B cells and also the more mature populations of B cells. Conversely, CXCR5 tends to be more restricted to the mature populations of B cells. So there is some state-dependent heterogeneity in this chemokine receptor expression. Now, if I plot CXCR4 on the y-axis as RAG1 on the x-axis, and let's say color it by IL-7 receptor, for example, you can see that there's this large double positive population of cells over here, showing us that there are populations of immature B cells that are RAG1 positive that are also CXCR4 positive. However, if I change the y-axis to CXCR5, you can immediately see that there's very, very little expression of double positive CXCR5 RAG1 positive cells, indicating at this point that I was expressing earlier that CXCR5 appears to be more restricted to the more mature populations that are CD20 positive. So this is just one of the functionalities that you can use to explore your genes of interest over here. Right, so what I'm going to do is I'm going to hide this. I'm going to go back to all the cell types, and I'm just going to change tack a bit. So the last thing I want to talk about briefly is looking at gene sets. So if we think about gene sets, I'm just going to use interferon gamma as an example here, because this is a gene that we're all very familiar with. So interferon gamma. Okay, if I expand on this over here, and if I color in interferon gamma, there appears to be some expression in this cluster over here, but it's not extremely clear to me because, of the high dropout rate of single cell sequencing. So what I'm going to do is I'm going to take my tool over here on the right histogram, and I'm going to drag only this portion over here. So what I've done here is I've only selected for cells where there is greater than log1 expression of interferon gamma. Again, I'm going to subset this population, showing us only cells that express interferon gamma at a high level, and the legends over here on the right. Now, if I color this in by cell type, then I can start to see which are the cell populations in my meningeal atlas or in the CNS borders that are expressing interferon gamma. And these are NK cells, CD8 T cells, and CD4 T cells. And also, you can also consider where the site of these cells are. So you can start to associate tissue and cell type associated differences in your genes of interest. Now, interferon gamma is just a cytokine, but let's say I'm interested in a whole collection of genes or a pathway that represents interferon gamma response, such as pathways we get from N6DB, etc., etc. So what I can do instead is I can create a gene set, right? So if I create a new gene set over here, I say I'm interested in interferon gamma response. I'm going to leave the description empty for now. And over here, let's say you have a collection of 100 genes or 50 genes or whatever that represent your pathway of interest. In the interest of time today, I'm just going to key in 5 or 6 genes. So for example, IFIT1, interferon gamma receptor 1, FI27, IF1, CXCR9, and CXCR10. For example, I'm going to create this gene set. And what this is going to do is going to score every single cell in this atlas for this gene set. So it's a bit slow today because of the internet speed. And I can color it in, and I can see which are the cell types that are responding to interferon gamma within my population. Again, I can subset it for the highest scoring cells, for example, if that's what I'm interested in. So going back to the entire atlas again, so back to all of this, there's one functionality in the top left corner here, which I will not go into in the interest of time today, because it does take some time due to its own computational intensity. But what you can do is you can also do differential expression gene analysis using this tool over here. Right, so that's all I'm going to be talking about for today in terms of the demonstration. Hopefully, the community will find this quite a useful resource for exploring your genes of interest, et cetera, and pathways of interest. But as mentioned before, if you've got any questions, do feel to reach out to us. We're very friendly, and yeah, we hope to be expanding this resource in the future. Thank you.
Thank you very much.